Okay. Something like that. Just tell me where you want to be. Okay. So how many of these do you guys know previously? Okay, cool. So this is a good chance for you to learn. You guys to learn. So let's start the next page. So here, I thought a lot of people would come, so I have to acknowledge my stupidity here because I'm still actually a middle schooler while doing this, and this is my first year of high school. So I really want to say that there's a lot of limitations to my presentations. There are furthermore researches that could be done and that I'm doing. So I will definitely focus more on these topics and give a probably even more detailed presentation in the future if someone wants to hear it. But this is like my acknowledgement of my stupidity. Next slide, please. So here, what is RNAi? So RNAi stands for RNA interference, right? So it actually won the Nobel Prize for 2006. Hey, thank you. It just started, so just sit down and listen. Yeah. Okay. So what is RNAi? RNAi stands for RNAi interference. It won the Nobel Prize in 2006. I pronounced it correctly. So Andrew Fine and Craig Mello has published a paper in 1998 related to the transfection or I don't think they use transfection, but they basically inserted RNA strands within the cell and observed the phenotypic changes. So this is generally epigenetics. What is epigenetics? Anyone? Do you guys know? What is epigenetics? Okay, cool. Epigenetics 中文名叫显性基因学。显性基因学 Okay, epigenetics is, the definition of epigenetics is the regulation of cellular characteristics by not altering the gene itself, such as histone modification, acetylation, methylation of DNA, and signaling pathways, and here, RNAi. So these are all ways to manipulate phenotypic characteristics of the cell by not changing the DNA itself, such as pathways generally. So it manipulates the expression of proteins within the cell. I'm going to explain this later, but basically, as what I talked about earlier, the siRNA joins the cell, joins a protein called disoprotein, and gets modified into a single strand, bonds to RISC complex, targets for the mRNA integrated. So this is the process of RNAi. Next page, please. So this is the general chart for RNAi. As you can see here, this is the RNA inserted, dicer-mediated cleavage, which the dicer just slice it into a few pieces, and it binds to RISC complex. Then it targets mRNA, cuts it, integrates it. This is an original chart from from Fires and Mellon's paper. So here there's something called twitching phenomena within C. elegans, which is the worm. So they normally do research on a few animals, and C. elegans is one of them. So here, the twitching effect within the C. elegans is actually genotypic, genotypics. It represents like the gene. So it's I actually don't know how to explain this, but basically, see here they injected the sense strand and the antisense strand, while all of them are wild types. So as you can see, okay, let's just look at the first category. When they inject the sense strand and the antisense strand, nothing happens. When they inject the sense plus antisense strand, which is the general structure of a siRNA, the C. elegans thus become strong twitchers. So this means that something is happening that manipulates the gene expression of proteins that leads to the twitching phenomenon within C. elegans. This means that there are some kind of regulation within the cell with this special when this special sequence or structure is inserted into the cell, or even produced by the cell itself. Here I want to emphasize the process of cell itself manufacturing or like transcripting these siRNAs to achieve regulations. So here we regulate the cell through inserting siRNA, but the cell itself produces siRNA, which regulates protein expressions. So this is epigenetics, right? It modifies protein expressions through not really changing the gene itself, but through more processes like this. Again, I want to mention that RNAi is highly conservative within animals, within living organisms, eukaryotes specifically. So it's common in plants even. I have met an iGem team that uses RNAi to basically act as a biopesticide. So when insects eat that plant, they actually die. Yeah, it's really cool. So here, this is the difference between mi, si, and shRNA. So here, miRNA is the thing that I mentioned that the cell generates within itself. So miRNA is basically just shRNA. mRNA is basically shRNA, but generated within the cell. So here, 就是统称是 siRNA。然后呢 mRNA 只是细胞内对于基因调控的一个特殊的一个 siRNA。So siRNA is just the sense and antisense strand, which is like the classical, like the normal picture that we see. But here, shRNA is this structure at there's this hairpin structure at the end. This is more stable. That's what I'm gonna say because I'm gonna mention the problems. This is more stable. Okay, next page, please. So this is the process of miRNA within the cell. So as you can see, there are miRNA sequences within the cell. Well, if they get transcripted, they get to this process of RNAi. At the end, protein synthesis or translation is like the mRNA is degraded, so translation doesn't happen. And this is cell itself regulating itself. Again, epigenetics. So here, CRC. So CRC is the main cancer that we want to approach. So for its anatomy, I don't really want to mention it specifically because there are much more to it if we want to extend it onto the kinds of CRC. However, it just grows on the colon and the rectum of the cat, like 直肠. So there are like 结直肠. It's basically like within your like 大肠, like the cancer. Why is it categorized? 本来有结肠癌和直肠癌. There's colon cancer and there's rectum cancer. Why do we classify them as the same? Because they have the same characteristics. And next slide, please. Wait, next slide, please. Oh, sorry. So this is the Hippo pathway within the CRC cells that leads to like its proliferation abilities. So this Hippo pathway is discovered within Drosophila in 2003. I pronounced it correctly. And TAE4 stands for TEA transcription domain 4. So as you can see, this is the basic structure of the signaling pathway. It's the pathway that controls proliferation and migration and apoptosis within the cell. So as you can see, the upstream signals actually contributes to the expression of YAP. So this is a main transcription coactivator. This coactivator bonds with TAD4, which is a transcription factor within the nucleus, and which achieves the expression of proliferation and apoptosis, like proliferation ability generally here. So there must be some change to this signaling pathway, which leads to cancer, right? Cancer divides really quickly. It doesn't, like apoptosis doesn't happen. Next page. So this is its general structure for the Hippo pathway. So as you can see, this is the final transcription coactivator that I just mentioned, YAP. It is like something called yes protein, like yes A protein or something like that. But it's basically a simple transcription coactivator that bonds with T84 within it. So there are multiple upstream signals like extracellular measured stiffness, GPCRs, which are hormone receptors, and cellular polarity, which is like the shapes of the cell, if I interpret it correctly. And then the energy stress, which will also activate and affect this pathway. Energy such as glutamine, which I listed above glucose. Glutamine is a major energy source for cancer, and cancer specifically adapts to glutamine through metabolic reprogramming. So all of these will contribute to the activation and the deactivation of the Hippo pathway, these upstream signals. So within cancer, there's definitely manipulations within these upstream signals. Next page, please. So why is it significant? Because we find, not we, but like a paper, according to Liu et al. From 2016, T84 actually is upregulated within cancer cells, meaning that T84 as a transcription coactivator, as a transcription factor, which is highly responsible for proliferation abilities of the cancer cells, is in a weird situation right now. It's not normal, right? So this abnormality leads to cancer cells, like directly. However, there must be YAP, right, which is a transcription coactivator. However, in 2016, a paper stated, I forgot the name, and but I forgot to cite the paper, but it basically suggests that YAP, T84 leads to proliferation on a YAP independent manner. So basically, if the T84 signaling intensity increase, the proliferation ability or characteristic of the cell will be directly affected without any other involvement of other parts of the cell. We could go through this really quickly. Next page. I'm not going to talk about this. Next page, please. Next page. Yeah. This is the CCKA test. Next page, please. Next page, please. Next page, please. I'm just gonna skip all of this. Yeah. Cool. Just skip all of yeah. Yeah, okay. Here, I really want to, this is a really important thing that I want to emphasize. Is mRNA dead? Is RNAi therapeutic dead? Therapeutic dead. Why do I mention this when I'm saying that we're doing this? Think of it. Think of it. Okay, next page, please. Next page. So, we're not even going to talk about this. Next page, please. Next page. Yeah, here. Other innovative approaches, yes. So here, cancer research is a broad topic to talk about because, so when I first heard of cancer, I thought of it just as a mutated piece of meat within the body. However, our average middle school interpretation suggests that there are definitely, so the cancer cells are way more deadlier than that somehow because it has proliferation, migration and invasion abilities at the later stages. So I want to extend on probably your interpretation, if this was you. Cancer the definition of cancer is a term for disease in which abnormal cells divide without control and can invade nearby tissues. Cancer cells can also spread to other parts of the body through the blood and lymph systems. So this is a really lame definition for cancer, but it really like defines all. So basically, the abnormal cell here represents the abnormalities within metabolism and within the cell. If that's a really general statement to say, but what gives a cancer cell its quick proliferation abilities? What gives its invasion abilities? Why does it differ than normal cells? It's because that it's metabolically it functions different than normal cells, such as the cellular respiration that normal cells can do. Normal cells use glucose as the main source of energy, but here, as I mentioned before, glutamine is used. The Chinese glutamine is guanzhuan, which is actually an amino acid. So metabolism is manipulated within the cancer cells to fulfill its energy needs, both energy, lipids. So there's lipid metabolic reprogramming, protein metabolic reprogramming, and like energy, basically energy stress, metabolic reprogramming, if that's a word. So this is other important approaches, innovative approaches. So think of this. If cancer were that different from normal cells, there would be a lot of approaches, right? Most areas. We can think of signaling pathways. We can think of proteins. We can think of upstream signals that controls the signaling pathways. And there are a lot more to it. But here, I'll generally say that signaling pathways and upstream signal regulations and proteins are some main approaches that modern scientists take towards cancer therapeutics.
Next page, please. Can you hear me?
Yes.
Just as something like a side thing, like considering this thing about metabolic reprogramming, would it be like a better solution typically if we have used the method of siRNA onto some metabolic pathway? For example, the use of glutamine, I think, yeah? Glutamine, okay. Glutamine in the pathway that results, that utilizes glutamine in cancer cells. If we targeted these pathways instead, could it potentially fix the problems that I talked about previously about ROS and stuff? Because it would be because we're taking off energy instead of blocking ways for it to cancerize, but blocking its life as a whole.
That is true. That is exactly true. That is absolutely correct. So there are a lot of approaches that we can take in, including the energy method. But for directly manipulating the metabolic patterns of cancer cells, it's so hard. There are so many things involved. There are so many interactions with the cell. So if I were to put pictures of the interactions of signaling pathways and different chemicals, it would be something this big and which is a protein or something linked to so many other proteins. So it's actually really, really hard to target one of them. But when I'm saying that glutamine, for example, it can't diffuse through the cell membrane easily. So we can manipulate its process for it to enter the cells, right? This is some projects that scientists are doing now by controlling the energy source of the cancer itself, right? So those are generally the innovative approaches and are definitely in research stage. Okay, thank you.
Sorry, but I think we have to end now. It's time.
Okay, it is absolutely fine. If next time, if I were to give this presentation again, which will involve ROS, I never thought that this ROS is that complicated. So when I'm doing this, I thought ROS just as a byproduct of metabolism and so on, but it actually involves a lot of process, okay, so here's like really crazy part. There's this ROS can lead to proliferation. ROS at the same time can also lead to apoptosis. How does the cell manage to do this? So there must be a redox level achieved. This redox level will be like, it could be achieved through manipulating oxidants, oxidants within the cells. So there's ROS, there's like reoxidants, proteins, which degrades the ROS through complex process. I don't even like want to mention it, but if we manipulate the oncogenetic expression, or even further on, the pathway itself, we can probably achieve this. So just a reminder, just